find that what kind of genetics is happening with their trait. So same thing was there with rust also. If we'll see the history, say at same time during 1905 to 10, heterosis phenomenon was explained by Searle. So that for that also he was trying to see that what kind of genetics is happening in maize. But for doing that, suddenly he discovered heterosis. Similarly, Biffen in 1905, what he found that yellow rust resistance, it is controlled by a single recessive gene, and the genotype was revert. So that genotype, what he saw, that it is resistant to yellow rust, and when he crossed it with resistant parent, the F1 was susceptible, and in F2 it was segregating in 3 susceptible and 1 resistant plant. So this was a very simple case of monogenic recessive gene, where the yellow rust resistance gene was controlled by single recessive gene. After that, people started searching what are the happening with other rust or other genes or the genes which is present in other genotype. So now, if you will see the one which is designated gene, we have 82 yellow rust resistance designated gene, 79 leaf rust resistance designated gene, and 60 stem rust designated gene. I am telling these are designated. Along with this, there are several other genes which are not designated. If you will go to China, they will not send their material to Australia or USA to get it designated. They have proved that their stock is having novel gene, but that is not designated. Even in India, the movement of genotype is restricted. Earlier it was moving freely, but after this Biodiversity Act, the movement of genotype is not easy. So for designation, you have to send your seed to Australia at PBI, where it will be given some particular number. So almost, if you will see, 250 genes are there for 3 rust resistance gene. But if these many number of genes are there, then why you need new genes? Second question comes, that if already we have these many genes, then why we need new genes? Already we have 250 genes, and that 280 for yellow rust, 80 for almost 80 for leaf rust, and 60 for stem rust. So if you will see the durability of these resistance gene, the basic native genes which is present in wheat, most of them are ineffective, and the one which is transferred from alien species, several of them also became ineffective, like your LR9, 19, 28, these are ineffective in India. We have virulent races against this, and the development of new pathotype in rust is very rapid. You will see that a variety is resistant, and if it is going to fill within 3-4 years, what you will find, that variety will become susceptible. Two things can happen, that new pathotype has developed, or already some pathotype was there which is not prevalent in pathotype population, but after starting large scale cultivation of that particular variety, that started, the frequency of that pathotype increased. That lead to susceptibility of that particular variety. So suppose you have some new pathotype, or sometime what you will find that altogether some new disease has been reported in particular crop. Like in wheat also, if you will see that blast has come. Suddenly it was not happening on wheat. Earlier there was some report, and then recently there is big report that blast is happening on wheat also. So for new disease or new pathotype which is causing disease, so what you need? You need a resistant source, so that your variety should be resistant. So if you need a new gene, so it can come from varieties which is already in cultivation, or obsolete varieties which was in cultivation, or land races, or in wild species. So suppose you need a new gene, first you should go to varieties which is under cultivation, or maybe under cultivation in recent time. So what is advantage if you are using varieties? They are the genotypes which is having good agronomic performance. So if you are getting any desirable gene from varieties, the yield penalty associated with that resistance gene will not be there, or chances are there that along with resistance you can select some other desirable trait also, because that is a popular variety which is having good agronomic performance and having good other agronomic traits also. If resistance is not available in varieties, then what should be your second option? Where you should go? Then you should go to land races. Why to land races? Because at least you will get completely fertile progenies. Your progenies will not be sterile. Like in wheat, you have cultivated variety and then land races. Both are Triticum acetivum having AA, BB, DD genome. If you will cross them, your F1 will be completely fertile. Where is the problem with land races? That land races will be having some inferior agronomic performance. So you have to remove those inferior traits which is associated with your land races, which you can do easily with the help of markers. By doing marker-assisted backcross breeding and by doing background selection, you can easily remove all those undesirable background effect which is coming from land races. Third, if your resistance is not available in land races also, then where you should go? Then you have to go to wild species. In wild species, you have three groups. One is primary gene pool, secondary gene pool and tertiary gene pool. If we will see in wheat, we have relatives in primary gene pool like we have hexaploid wheat like Sphaerococcum having similar genome constitution. You have Spelta that is also having similar genetic constitution. If you will go to little bit far, even Durum and Dicoccum are member of primary gene pool, or the species which is having similar genome and even they are diploid, like Triticum monococcum is diploid having AA genome and Toschai is having DD genome. Okay? So, they will also be in primary gene pool, although if they are not having similar ploidy level, as your bred wheat is hexaploid and tetraploid wheat is Durum and Dicoccum is tetraploid wheat and your Toschai and monococcum is diploid wheat having two genome only, but they are having complete pairing. The A genome of monococcum or uratu will pair easily with your A genome of wheat. In F1 you will get sterility, but once you are making one or two back cross, you will recover complete fertility. So, you do not need any extrapolation using any cytogenetic technique. Now, if it is not available in primary gene pool, then where you will go? You will go to secondary gene pool. Secondary gene pool means like your Triticum timophoebe. What is genome of timophoebe? AAGG. So, one genome is common with bred wheat. AA is common genome and G is having homology with B genome. So, if you want to transfer a gene from A genome, it do not need any cytogenetic technique, because A genome will pair from both the species and crossing over will occur between A genome of timophoebe and A genome of wheat and transfer will happen. But if you want to transfer a gene from G genome, in that case you have to induce homologous pairing. That can happen through using NALI5B. What is NALI5B? All of you must be knowing. 5-BLPH is a gene which is responsible for stopping your homologous pairing. If you are removing that gene using NALI5B, it will induce homologous pairing. In that case, what will happen? The pairing will occur between B genome and G genome and your transfer will occur from G genome also. Third, if it is not available anywhere, then you should go for tertiary gene pool. Tertiary gene pool means they are not at all related genome, like your rye. It is not homologous to any of the genome. Or you have some wild species like Agilops margraffi, chordata also sometimes you will find. So, they will write CC genome. So, if you want to transfer some gene from CC genome to bred wheat, you need to induce homologous pairing, or sometime what people will do, they will do some mutagen treatment also, so that transfer should occur. Now, even if it is not available anywhere, then some other approaches are there, like your transgenic. So, suppose you identified a gene that okay, now my desired gene is present in this particular genotype. So, before using that gene, we have to understand what is basic nature of that gene. That will decide what kind of a strategy you should adopt to transfer or to use that gene in your breeding program. So, this is most basic thing. I am sure that all of you must have read in BSc or somebody would have discussed. So, what is genetic analysis? For doing genetic analysis and why we are doing genetic analysis. Genetic analysis is done first to understand what is the genetic nature of resistance gene, whether it is a dominant gene or it is a recessive gene. Second thing, to know the number of gene which is responsible for that resistant phenotype. How many gene is responsible which is leading to that particular resistance. Suppose that is a recessive gene, small r, and that is only one. So, only one small r. So, how it will help? This will help you to design what kind of breeding strategy you should use. Suppose there is a monogenic recessive resistance gene. What will happen in F1? All the plant will be susceptible. Now, if you want to transfer it through backcross breeding, okay, your recurrent parent will be having capital R, which is responsible for susceptibility. What will happen in BC1 F1? All the plant will be susceptible. So, if you are not knowing that what is the nature of your resistance gene, you may not be able to detect whether my backcross population or my F1 population is having resistance gene or not. Maybe that is self-driven or maybe some outcrossing has happened. So, you will not be able to decide, but if you are knowing that my resistance gene is recessive, so what you will predict? My F1 will be susceptible if it is true cross. And if you are making backcross with your recurrent parent which is carrying capital R, it is predicted that all the individual will be recessive, all the individual will be susceptible. In that case, what you have to do in backcross breeding? Either you have to self for one generation to BC1 F1 and you have to identify homozygous resistant plant, and then those individual will be backcross to your recurrent parent if marker is not available. If you have molecular marker linked with recessive gene, you can identify those. You are not having any board, I think. If you have molecular marker, so with help of molecular marker you can identify, even though the plant is susceptible, among susceptible what are the genotypes or what are the individuals which is carrying my recessive resistance gene in heterozygous condition. So, you can design your breeding strategy. Second thing, suppose it is a monogenic trait and if you are transferring it through backcross. So, out of 4, how many individual will be resistant and how many will be susceptible? Okay. So, if it is monogenic, so even in a small population you will get some desirable plant. If it is polygenic, you need to have a large population to catch all the genes. So, again two things will be decided, what should be the strategy and what should be the population size, that will be decided by basic genetic nature of a gene. Now, second thing will come, how to do genetic analysis. This is very simple. You have to take two parents, the parent where you have identified that you have a resistant gene, and then you have to take a susceptible plant. You have to cross them, you will get F1, then self-dose F1. There are some precaution required which I will tell while discussing mapping population development, and here also I will discuss little bit. So, then you will self-dose F1 plant, you will get F2 individuals, and then again you will self F2 individuals and you will get F2 derived F3 lines. Then after development of these seeds, what you will do? You have to do phenotyping of your F1, F2, F2 derived F3 and test cross. What is test cross? You are crossing your F1 plant with your recessive parent, not resistant parent, recessive parent, and recessive parent can be resistant or susceptible. That will be decided by what is the genetic nature of gene. Then after phenotyping your F2, you have to do chi-square test. That is simple chi-square test, and you will test goodness of fit, that in which ratio it is fitting. If it is fitting in 3:1, 3 resistant and 1 susceptible, that is controlled by 1 dominant gene. 1 resistant, 3 susceptible means 1 recessive gene. Then what are the precautions which you need to take while developing these populations? First thing, you should always maintain purity of your line. Why I am telling this? Resistant parent you have identified, that to X line you want to see that what kind of genetic nature is there for this resistance gene. So, in that also sometime what you will find that there is some variation or there is chances of some mechanical mixture. So, people will think, or if you are telling to your student just go and make cross, he will not assure that whether that plant is pure or not. So, if he is making cross on some off type plant, your genetic analysis will not be successful. So, before doing any genetic analysis for rust resistance, what we are doing in glasshouse, we are testing those seedlings on which we want to make cross. Okay? We are taking resistant parent, we are taking susceptible parent. First we will test. If it is seedling resistance gene, we will test in seedling reaction only, that whether resistant parent is resistant or not and susceptible parent is susceptible or not. Sometime what will happen, I have seen this, it is happening most commonly, that they will tell Agra local is susceptible, you make cross with Agra local. Or if you are making Durham, they will tell take some local or whatever, like Thatcher, they will tell you take Thatcher, you make cross with resistant parent, your F1 will, you will get a mapping population. But suppose there is some mixture in Agra local and some resistant plant came. Okay? So, your F1 and F2 will not be a cross of resistant and susceptible, maybe that is cross of some other thing. So, it is always desirable that if you are doing genetic analysis, assure that your resistant parent and susceptible parent are resistant and susceptible. There is no mixture and they are true to the type. Got my point? Maybe that is resistant, but that is not my stock. There is some mixture. So, you have to assure that this is my stock for which I want to do genetic analysis. Then should confirm hybridity. Sometime what will happen, that you have selfing or some outcrossing is happening from some third line. So, you have to confirm hybridity, that this F1 plant is product of my resistant parent and susceptible parent, that is not selfed one and that is not because of some outcrossing. So, that you can do with the help of molecular marker. Although wheat is a self-pollinated crop, so selfing will take place automatically, but even 2 to 3% crossing is reported. So, if you are developing a mapping population, it is always desirable that you should do a strict selfing by covering those spike of your F1 plant. Suppose 10 spike is there, so you should cover all 10 spike and then you should take only those strictly selfed plant. Okay? Fourth thing which comes in genetic analysis, you should always use pure inoculum. If your inoculum is contaminated, your genetic ratio will be changed, or sometime you will feel noise is there. You are not getting expected segregation. Okay? How? I will discuss little bit. Now you come to case. I will start with some simple case and then we will see what is happening. Suppose the stock where you want to do genetic analysis, that is having a monogenic dominant. I am telling you result. This will come at last. So, what will be the phenotype of F1? F1 is resistant. F1 is resistant means your resistant line is carrying one dominant gene. It is possible that there is some other gene also, maybe 2-3 dominant gene is there, or one dominant, one recessive gene is there. But if at least among all the genes, one gene is dominant, then your F1 will be resistant. Now, if you will see segregation in F2, if they are segregating in 3 resistant and 1 susceptible, it will confirm that that dominant gene is monogenic, that is single gene which is responsible for resistance. And then once you will go to F2 derived F3, F2 derived F3, what you will do? You will self all the F2 individual which you have generated from F1 plant, and then what you will do? You will take 10 to 15 seed, that is sufficient for your monogenic dominant case. So, 10 to 15 seed you will put in a tray. If time permit, today I will try to show you how we are doing this in glasshouse. So, 10 to 15 seedling of single F2 plant you will put in tray. You got my point? Suppose you have 400 F2 families, so 400 row of F2 families you will plant, and for each family you will use 10 to 15 seed and you will get around 10 to 15 seedling. So, you will be having 3 class in that F2 family. 1/4 will be homozygous resistant. Homozygous resistant means capital R, capital R. So, all the 10 to 12 or 10 to 15 seedling in that family will be resistant. It means this is product of that F2 plant which was having capital R, capital R. Then what you will find, some of the families are segregating again in 3:1. Out of 15, 3 to 4 you will find that susceptible seedling is there, and then rest are your resistant. So, those are segregating families. And then third class you will find that all the plants are susceptible, that is called homozygous susceptible. So, in F3 family you will find 3 kind of families. One will be homozygous resistant, 2 frequency of 2 will be segregating, and 1 frequency will be homozygous susceptible. Now, what is there to take care? That recessive class, like in your monogenic dominant case, what is recessive class? Susceptible is recessive class, small r, small r. So, in F2 that recessive class, in F2 it will segregate in 3 resistant and 1 susceptible. So, what you will see in F3, that susceptible plant which has given F2 derived F3, all will be homozygous susceptible, because that is product of small r, small r. Sometime this recessive class can also be used in mapping, okay, because you cannot distinguish resistant class whether it is homozygous or heterozygous. People will use recessive class of F2 family only for mapping. Now, coming to test cross ratio. In test cross, we will cross with recessive parent. So, your recessive parent is small r, your F1 is capital R, small r, and it will segregate in capital R, small r, which is resistant, and a small r, which is susceptible. So, it will give you a ratio of 1 resistant and 1 susceptible. So, again it will confirm that this is a monogenic dominant gene. Okay? This is just a validation. First, in F2 it has segregated in 3:1, it has indicated that this is monogenic dominant reaction of F1, or resistance in F1 has indicated this is dominant. In F3 it has segregated in 1:2:1, again it has confirmed that this is monogenic dominant, and last in test cross population again it has segregated in 1:1. Here your heterozygous will be resistant and homozygous will be susceptible. It has confirmed that this is a monogenic dominant. Now, what will happen if you have monogenic recessive trait? What will be the phenotype of your F1? F1 will be susceptible. F1 is susceptible means your gene of interest is recessive. Now, this can happen that there is one recessive gene or two recessive gene, but one thing is sure, that your gene is recessive. Now, if you are coming to F2, if it is segregating in 3 susceptible and 1 resistant, it means this is monogenic recessive. Now, coming to F2 derived F3, the individual which you have harvested from resistant F2 individual, all those F3 families will be homozygous resistant. Why? That is a recessive class. Small r is responsible for resistance, and in F2 itself, if it is resistant, means that is having small r, and in F3 they will not segregate, that will remain resistant. Then you have capital R, small r, which will segregate in F3 again in 1:3 ratio, and you have some homozygous susceptible plant, which is capital R, capital R. In back cross, if you will see, again you have two genotype, one is heterozygous and one is homozygous. Here your heterozygous will be susceptible and your homozygous will be resistant. This is different from your earlier case. There it was vice versa, your heterozygote class was resistant and homozygote class was susceptible. Now, what will happen if more than one gene is responsible for your resistance? These all are genetic analysis. It will, once you will start working on rust or any disease, if you will start working, these are the basic step which you have to follow. You have to make cross between P1 and P2, you have to develop F1, you have to see reaction of F1, what kind of reaction it is giving. Then you will develop a F2 population, then you will see what kind of segregation is there. Then you will develop an F2 derived F3 population, you will confirm it, and then you will also confirm in test cross population with your recessive parent. Now suppose more than one gene is responsible for your resistant. Take case one by one. If you are having two dominant gene which is responsible for resistance, in that case what will be the phenotype of F1? F1 will be resistant. Capital R1, small r1, capital R2, small r2. So that individual will be resistant. If you are going in F2, it will segregate in 15 resistant and 1 susceptible. What are the individuals which will be resistant? All those individuals which is carrying dominant allele even at one locus, maybe in homozygous or heterozygous condition. So only one genotype is left which will be susceptible, small r1 and a small r2, small r2. Rest all 15 individual will be having at least one dominant allele for R1 or R2. So those 15 genotypes will be resistant. If we will go to test cross ratio, it will segregate in 3 resistant and 1 susceptible. How? This is your susceptible parent which will be having a genotype of small r1, small r1, small r2, small r2. What is genotype of F1? Capital R1, small r1, capital R2, small r2. It will give you 4 types of gamete: capital R1, capital R1, capital R1, small r2, small r1, capital R2, small r1, small r1. If you will cross them, you will get these 4 genotypes. So what are the individual which will be susceptible? Only this individual will be susceptible, which is homozygous recessive for both the genes. Here you have both the genes, both the allele in dominant form. Here you have one, here you have one. Only 4th individual is there which will be susceptible. So it will segregate in 3:1 ratio. Now what will happen if one dominant and one recessive gene is there? The F1 phenotype will be resistant. Why? Because one dominant gene is there. That will lead to phenotype of your F1 plant. I told if F1 is resistant, it means at least one thing you can show, that one gene is there which is dominant. Maybe other gene will be there, but at least one is there which is dominant. Now if you will see segregation in F2, you will get 13 resistant and 3 susceptible. What are those 3 susceptible individuals? Suppose your R1 is dominant and a small r2 is recessive gene. So if single plant is carrying both the gene, what will be the genotype of your resistant plant? Capital R1 and a small r2, Okay, that will be the genotype of your resistant parent. If you are crossing it with susceptible parent, what will be the genotype of susceptible parent? Small r1, capital R2. In F2, it will be heterozygous for both the gene. And which 3 individual will be susceptible? Which 3 individual will be susceptible? Small r1, capital R2. This homozygous individual will be susceptible, and 2 small r1, capital R2, this will be having frequency of 2 individual. That will be susceptible. So total out of 16, 3 individual will be susceptible, rest 13 will be resistant because of one locus or other locus. If you are going to test cross, which individual will be susceptible? Only this will be susceptible. Why? Because here you have small r1, here your dominant locus is lacking, and here this recessive locus is suppressed by a dominant locus. So these 3, this will be resistant because of R1, this will be resistant because of R1 again and R2 also, and this will be resistant because of R2 locus. But here this will be susceptible. In earlier case, you are having these 3 resistant and 1 susceptible. In second case, you will be having this resistant and this resistant. This will be susceptible. So this is the case if it is one dominant and one recessive gene. Now if both are recessive, can happen that 2 genes are there and both of them are recessive. In that case, what will happen? your F1 will be susceptible. What will be the phenotype of F1? Capital R1, small r1, capital R2, small r2. And what is genotype of your resistant parent? Small r1, small r1, small r2, small r2. You are crossing with susceptible parent which is having capital R1, capital R1, capital R2, capital R2. F1 will be heterozygous for both the locus and your F1 plant will be susceptible. And once you are developing F2 population, 7 plant will be resistant and 9 plant will be susceptible. Okay? What are those 9 plants? They are either heterozygous at both the locus and you have one dominant allele at both the locus, maybe in homozygous condition or heterozygous condition, but dominant allele should present at both the locus. So those 9 individual will be susceptible, and the one which is lacking dominant locus, even at dominant allele even at one of the locus, that individual will be resistant. If you will go to back cross, again you will find 3 resistant and 1 susceptible. So here this will be susceptible and these 3 will be resistant. Okay? So in test cross, in all 3 cases you will get segregation in 3:1. But in F2 you have different segregation ratio. So by this you can tell that whether your gene of interest is dominant, recessive, and what is the number of gene. Now I told you your pathotype should be pure, then your genetics will be clear. I will give you one example, that how this composition of pathotype will decide your genetics. In leaf rust we have many dominant genes. I have taken example of 2 dominant gene, LR19 and LR28. Suppose one resistant individual is there which is carrying homozygous LR19 and homozygous LR28. So what will be the genotype of your resistant plant? LR19, LR19, LR28, LR28. So this is a simple case of digenic dominant. Okay? Your F1 will be heterozygous for both the locus. If you will see F2, this will be homozygous for both the locus LR19 and LR28. This will be homozygous for one and heterozygous for other, but dominant allele is there. Like this 16 will be having. All of you must be clear about Punnett square, I am sure, that how to draw this. And then this will be recessive for your homozygous recessive for both the genes. Now suppose you have taken a pathotype 77-5 which is a virulent to both the genes LR19 and LR28. So what will be your ratio, genetic ratio in F2? You will get 15:1. So here all this 9 individual is giving resistance because both the allele is present in dominant form. In these 9 individuals, if you will see, at least one dominant allele will be there. Here it is LR19, here it is LR28. Both the alleles are there here in dominant form. Here one gene is there in homozygous form. Here another gene is in homozygous form. In these two, if you will see, one dominant allele is there. Here LR19 is in dominant form, here LR28 is in dominant form. So 15 individuals are there which is having one or other allele in dominant form, that will provide you resistance to pathotype 77-5. Now you change your pathotype to 77-8. 77-8 is a pathotype which is virulent against LR19. So what will happen? All those individuals which are carrying LR91, only LR91, even if it is dominant, like this. Here only LR19 is present and that LR19 is not effective against 77-8. So this individual will be susceptible. Here you have only LR19 in dominant form, it will be susceptible. Here you have only LR19 in dominant form, this will be susceptible. So you have 6, 8 and 14, 16. 6, 8, 14 and 16. These 4 individual will be susceptible. So out of 16, 4 is susceptible and 12 is resistant. So what is the ratio? 3:1. So just by changing a pathotype, 15:1 changed to 3:1. Now you have used another pathotype 77-10. This is virulent against LR28. So, all those individual which is carrying only LR28 in dominant form like this 15, 12, 16 and this one 11. These 4 will be susceptible. Again it is 3:1. So, if you are not screening with 77-5, you cannot tell that 2 genes are there. If you are screening only with these 2 races, you will tell that single gene is there and that is giving 3:1. You cannot tell that they are 2 different genes. Okay? So, simply by changing pathotype, your segregation ratio will change. So, it is always desirable that you should use at least 2 pathotypes for your genetic analysis confirmation, and you should take most prevalent one. Then you can tell exactly, suppose I have taken 77-5, then 2 race is there, I can tell that this genotype is carrying 2 resistance gene against 77-5. But if you are keep on changing pathotype, your resistance will change. Now, sometime what will happen? People by chance they will mix 2 pathotypes. Suppose you have mixed 2 pathotypes 77-8 and 77-10. So, wherever you have pyramided form means 2 dominant allele is there, that will be resistant, because against both the pathotype you have dominant resistant allele. But the moment any one of them is lacking, plant will become susceptible because of virulent pathotype. Here you have LR28, so mixed one will be susceptible because of 77-10. Here you have LR19, this individual will be susceptible because of 77-8. So, you will get a different ratio and you will get confused, and in a scoring plant also you will find some difficulty. Suppose you are using 77-5, all the plant will give a uniform kind of pattern for your scoring like FLEC 1 or whatever you are getting. The moment you will mix 2 pathotype, in single plant what you will find, like here only what you will find, that there are some 3 kind of pustule, as well as what you will find there are some flecks. So, that will mislead you that whether that is susceptible or X kind of reaction which is happening. So, you should always use pure pathotype if you are doing genetic analysis, especially for seedling resistance. You can do for APR with little bit precautions. Now, you came to know that your resistant stock is carrying some resistant gene, whether that is monogenic, digenic, that you have proved, or you came to know that that is carrying a dominant gene, a recessive gene, single gene is responsible, 2 gene is responsible. Now, this can happen that as we discussed, 82 yellow rust resistance gene are known, 79 leaf rust resistance gene are known, or 60 stem rust resistance gene are known. My stock is carrying one of that gene. Okay? So, already this is known, that is nothing new. So, how to prove novelty of your stock, that your stock is carrying some novel gene? So, the basic way to predict your gene is that what people will do, that they will see pedigree of that particular genotype. Suppose your genotype X is there and you have used transfer as one of the parent in development of that line. Transfer was the stock which was developed from Umbellata introgression, which is having LR9. So, you can predict that my stock is carrying LR9. So, by seeing pedigree you can predict that this may be the probable gene which my stock is carrying. If you are not sure, or some land races is there for which we do not know pedigree, or we are not knowing what kind of gene it is carrying, then you should go for second stage. If you will see, first we will identify whether my stock is carrying resistance or not. Initially, we will use 1 or 2 most prevalent races, the one which is most predominant. If it is resistant to that predominant race, then you will go for multi-pathotype testing or single race testing in isolation. You will test with 20, 30, 40 pathotypes, whether my stock is carrying resistant to all the pathotypes or it is effective only against few pathotypes and it is susceptible against few pathotypes. Somebody has taught gene postulation? Gene postulation has been taught. So, based on your SRT data, you can postulate that. Suppose I will take one simple example, as I told for LR9 only. In India, we have only one pathotype which is virulent to LR9, that is 77-7. If my stock is giving resistant to all the pathotype, but it is susceptible to 77-7. So, we can predict that my stock is also carrying LR9. So, second step is based on gene postulation, you can predict that my stock is carrying this particular gene. Third thing, if you are not clear even after gene postulation whether my gene is novel or not, then you have to go for some extensive crossing program. Initially only few resistance genes were known, so understanding allelic relation was easy. Suppose you have only 5 resistance gene known for a particular disease, you have to make cross with only 5 stocks which is carrying, suppose LR1, LR2, LR3, LR4 or LR5. We are identifying LR6, so I have to make cross only with these known stock. Now we have 79 LR genes. So it is not possible to cross with all 79 genes and then to see whether they are different or not. In that case, what will be desirable? That we should know that what is location. I will come to that. If we will see, if your known stock and if your unknown stock for which you want to test whether it is carrying a known gene or not, and you have one known stock, if they are carrying 2 different gene, what will happen? Suppose this is known gene, R1 is known gene and my stock is carrying R2. Okay? So, what will be the genotype of your known one? Capital R1, small r2, small r2, and my stock is carrying R2. F1 will be heterozygous for both the locus. Okay? If it is segregating in 15:1, it means my stock is carrying a different gene than the one which is carried by a known stock. If 4 genes are known, you have to cross with all 4 known stock, and if they are segregating in 15:1, if both are dominant, then we can tell that my gene is different from earlier 4 known genes. Okay? Now second case can come, my stock is carrying same gene as of known. Okay? My stock is also carrying capital R1 and the known one is also capital R1. F1 will be resistant, so based on F1 you cannot distinguish. Here in F2 what you will find? You will not find any susceptible individual. It means they are carrying same gene. Now new thing will come in your picture. This can happen that the 2 genes are very closely linked and you are screening small population. Okay? In that case what will happen? You are not getting any recombinant gamete. If you are getting recombinant gamete, then only you will get some susceptible individual. If they are independent, they will segregate in 15:1. If you are not getting susceptible individual, it needs again confirmation in large population. So you have to take a large population and then you have to see that whether we are getting any susceptible plant or not. That will be frequency of susceptible plant will be decided by how closely linked those 2 genes are. You are getting my point? If I don't know, in board I can explain you better, or you just imagine how it will happen. Your genotype is capital R1 and a small r1. They are present on same chromosome in close vicinity. Your known stock is capital R2 and small r2. That is also in close vicinity. F1 will be heterozygous, but they are very close. So, all the gametes or most of the gamete will be capital R1 and small r2, or small r2 the second one. And in F2 all the individual will carry at least one dominant. But if crossing over is taking place between them, then only you will get a gamete of small r1, and that will give you a susceptible individual. So, if you are getting few susceptible individual in large population, then you can tell that my two genes are different, but they are very closely linked. Fourth case will come that the same gene can have different allele. So, this can also happen that my stock and unknown stock is carrying two different allele of same gene. This is my unknown stock capital R1, and this is known stock. F1 will be capital R1 and capital R2. They are carrying two allele of same gene, one which is from my unknown stock and the one from known stock. F1 will be resistant and all the F2 will be resistant, like this one. But then how you will tell that this is different allele, this is not same allele, based on multi-pathotype testing. If they are same allele, you are testing with 40 pathotypes, they will give similar reaction. If they are different allele, out of 40, or you take 5 pathotypes, if it is giving resistance to 1, 2, 3, 4, first gene, second gene is giving resistance to 1, 2, 3 and 5, it means they have little bit different resistance pattern. But you are not getting any susceptible individual in F2. It means they are two different allele of same gene. Second thing, which pathotype you have to use for this here to confirm they are same allele. We have to use one pathotype which is avirulent to both the alleles. You are getting my point? If you are using, suppose pathotype 3 is virulent to R1 and avirulent to R2. So, this will segregate in 3:1. If you are using another one which is virulent to R1 and avirulent to other one, again you will get 3:1. But if you are using a pathotype which is avirulent to both the alleles, you will get all resistant plant. Even in very large population, if you are not getting any susceptible plant, then you can tell that these are two different allele of same gene. Okay? But ultimate confirmation for this allelic relation will come sometime. What people has found that earlier they have reported that these two are different allele of some gene, but there was one independent loci which was interacting with that gene that has been responsible for this pathotype difference, pathotype disease resistance reaction difference. So, ultimately it will come once you will go to sequence level and you will be able to confirm that this is happening because of my sequence, or in several crosses with several pathotype they are giving same reaction. In Rust you will find that for some of the genes they will write like LR4A, B, C, and A, B, C, D they have written, and later on they told these two are not different allele, they are same allele. Okay? This they have explained in not one book is there, not. That is why Douglas Arnott, that book if you read, he has explained this very nicely, that how to do this test of allelism to confirm that whether they are carrying same gene, or they are two independent gene, they are two linked gene, or they are two allele of same gene. Now, as I discussed, you have 79 leaf rust resistance gene, and you want to confirm that whether your stock is carrying novel gene or not. So, if you are going through conventional process, what you have to do? You have to make cross with all LR79 leaf rust resistance gene, and then you can tell that I have ruled out all this L79 rust resistance gene, my gene is new. But if you are knowing that what is location of your resistance gene. Suppose with help of monosomic analysis or with help of molecular marker, we came to know that my resistance gene is located on some particular chromosome. I'll give you one small example. After mapping in one of the stock, we came to know that our resistance gene is located on 1DS, small arm of 1D chromosome. That arm is already carrying 3 leaf rust resistance gene, LR21, LR42 and LR60. Now we have to prove that our stock is carrying some different resistance gene compared to your LR21, LR42 and LR60. First thing what we will do, we will go for multi-pathotype testing of our stock along with LR21, LR42 and LR60. If there is some variation, like LR21 is susceptible to so many races, then we can tell that LR21 and our stock is different. Maybe they are carrying different allele or different gene that we can test only after doing test of allelism. Suppose your stock and LR42 is giving resistance to all the pathotype. In that case, you have to prove it through test of allelism in F2 only. If you are getting some susceptible individual, then you can tell that my stock and LR42 is different because I got some recombinant, and then in F2 we are getting some susceptible individual. Same thing will happen with your third gene also. So this is one of the reason, and second thing, you need to have molecular marker if you want to transfer your resistance gene rapidly and in more precise way. You want to remove all the linkage drag and you want to recover maximum background of your recurrent parent. Then you need to have molecular markers and you should map that marker with your gene of interest. So for any molecular mapping program, you need to have 3 things. First is your mapping population, second is your molecular marker, and some software is required by which you can do marker trait association. Then what are the steps which is involved in molecular mapping? First is development of mapping population. You will take your contrasting parent. Suppose here your purpose is to map leaf rust or stem rust resistance gene. You take resistant stock, cross it with susceptible stock, and then you develop a mapping population. After development of mapping population, what we'll do? We'll do phenotyping of that mapping population. Then we'll go for genotyping of mapping population, and then we'll see marker trait association, whether or which gene or which marker is linked with your gene of interest. So what is a mapping population? Mapping population is a population that is suitable for linkage mapping of genetic markers, okay? And mapping population can be generated from 2 parent or multi-parent. You have biparental mapping population like F2, F2 derived F3, backcross population, double haploid, RIL or NILS. You have multi-parent population also like NAM population or MAGIC population. But if you will see rust resistance gene, in general most of the paper you will find they will use F2, F2 derived F3 or RILs or NILS they will use for fine mapping for rust resistance, and most commonly if it is monogenic, F2 derived F3 and F2 will solve your purpose. Okay. So what is F2 population? This is most simplest population and it will take minimum time. Somebody has discussed all these mapping population, then I can escape. Okay. So you have 2 parent, parent 1 and parent 2, one is resistant parent, one is susceptible parent. You cross them, you will get F1, you self that F1, you will get F2. Again, as I discussed earlier, you should always take care of purity and true to the plant. Okay, if you are targeting your stock should be true to the type. There should not be any contamination. That will hamper your mapping program. Then this will undergo only one cycle of meiosis. You will be having if codominant marker is there, it will segregate in 1:2:1. If dominant marker is there, it will segregate in 3:1. Now, what is the thing which you should take care while doing mapping from F2 population? Suppose you have parent 1 and parent 2. So, you have to emasculate one parent, and from another parent you have to take pollen. So, the plant which you are using for emasculation, some other tiller will be there, you self that plant and collect leaf from that plant itself. Even your line is homozygous, it can happen that within a variety also there is plant to plant variation that can hamper your mapping program. So, the plant which you are using for mapping, that if you are using for your parental polymorphism, that is best. If somehow you have lost, then you can take, but you should always take care of your purity. Second thing, again pollen parent also you should collect leaf from same plant from which you are taking pollen. So, in single spike you will get 20 to 30 F1 seed. Each F1 seed you will sow, and then you should confirm hybridity of each F1 plant, and then strictly you self those plants, and always maintain identity of those F2 seeds. Suppose from plant number 1 you got 400 seed, plant number 2 you got 400 seed. So, you should always maintain that this 400 plant is from my first F1 plant, second 400 seed is from second F1 plant. If you are mixing, chances are there that some error will happen. Okay. So, what is merit of your F2 population? It will take minimum time. You can do some preliminary mapping, and it requires least time. Only one season for crossing and one season for selfing, you will get your F2 seed. So, F2 seed you can grow and you can screen it for seedling resistance, or if you want to see for field resistance, you can put it in field and you can screen for field resistance. So, what is some demerits or limitation of this mapping population? This is immortal population, means if you are using this 100 F2 seed, same 100 F2 seed you will not get. If you are using another 100 F2 seed, that will be different genotypically from this 100 seed. Segregation ratio you will get similar, but this is not same 100 F2 plant which you first 100. Then difficulty is there in polygenic resistance. If the resistance is controlled by polygenic trait, their mapping will be difficult. Already I have discussed that these three things you should take care. Okay. Now, you developed an F2 population. Suppose you have taken 400 F2 population. So, you self all those 400 plant, what will happen? You will get 100 to 200 or 300 seed from each F2 plant. So, each F2, the seed which you are harvesting from each F2 plant, that is giving you F2 derived F3 individual. If it is monogenic dominant, that will segregate in 1:2:1. One will be homozygous resistant and one will be homozygous susceptible. In rust resistance, if you will see, F2 derived F3 population will serve your purpose. What you should do? You develop an F2 derived F3 population, take homozygous families, take homozygous resistant, take homozygous susceptible, and then you go for bulk segregant analysis, or if you wish to go for some other method, you should go for that also, because here you are knowing that there is no heterozygous involved. I will discuss little bit what will happen, what complications are there if heterozygous individual will be there. So, this will be, I feel, ideal population for your mapping if your stock is carrying single dominant or recessive rust resistance gene. Then your double haploid population. How to develop double haploid population, all of you must be knowing, either by anther culture or ovule culture, or there are some techniques by which you can, like bulbosum technique you have, or in maize you have some technique like in vivo haploid inducer induction is there by using haploid inducer lines. So, you induce haploidy, and then by doing colchicine treatment you will get doubled haploid population. What is advantage with doubled haploid population? In minimum time you will get completely homozygous plants and you will be having a homozygous population. This will be an immortal population. Immortal population means this you can use forever and you can replicate at several places, or same population you can use for testing against different pathotypes. This can be replicated. That is why we are telling that this is immortal mapping population. Here you will get homozygous line very rapidly. But what is disadvantage with double haploid? Either you need some special stocks like haploid inducer line which is not available in all the crop, or you need a facility of tissue culture to develop double haploid line through pollen culture or ovule culture. Second thing, if protocol is not standardized, getting large population is difficult. In India or at some places where sophisticated facilities are not available, what you will see, doubled haploid population has been developed, but the population size is very less. Okay. In maize we have very good haploid inducer line, there you can develop a large DH population using haploid inducer line. Second thing, what you have to take care, that there should not any selection happen for development of any immortal population, whether it is DH or RIL. Here if your purpose is to develop double haploid population, you should go for haploid induction on F1 plant. Okay? You cross P1 and P2, you will be having F1 plant. If you want to do pollen culture, take pollen from F1 plant. Okay? If you want to do ovule culture, take ovule of F1 plant. Or if you want to do it by haploid induction, induce haploid on F1 plant itself, P1 into P2 F1. F1 you cross with haploid inducer line, you will get some haploid embryo containing seed that you treat with colchicine, and then you will get DH population. If you are going to F2, your segregation will be distorted. Suppose you are trying to map capital R1 and capital R2 capital R2. F1 will be heterozygous for both the gene. So, you will get all 16 combination in F1 plant. But once you are coming to F2 plant and you are selecting any one of the F2 plant, you will see segregation which is happening because of that F2 plant only. Okay? So, if your purpose is to develop double haploid mapping population, you should use F1 plant only. If your purpose is to develop some superior line in breeding program, then people will recommend that you should go for one cycle of selfing and take best F2 plant, and then you induce this haploidy and then you convert double haploidy, so that you will get best individual. The poor individual will be discarded in F2. So, these two purposes will decide which individual we have to select in during course. Then another your mapping population is your back cross mapping population. So, you cross with your recessive parent, okay? You will be having different types of individual in BC1 F1, and then you can generate BC1 F2 also. But if you are crossing with resistant parent, then you will not find any segregation in BC1 F1. Suppose you are crossing with capital R, all the plant will be resistant. This you have to take care that you have to cross with recessive parent only. There are some very good advantages with your back cross population. First thing, it need less time as of your F2. You back cross F1 with your recurrent parent or parent, you will get a back cross population. Second advantage what it will give, suppose you want to do marker-assisted introgression of your gene of interest. So, you have generated BC1 F1, 50% of the plant will be resistant and 50% of the plant will be susceptible, that you can use for mapping, and then you retain those resistant plant and give one more or two more back crossing your gene will be transferred in your desirable background. So simultaneously you can do mapping as well as you can transfer your gene of interest in some desirable background. But the biggest limitation with your backcross population is that you have to make backcross. So the size of population will be limited if pollination is difficult. If emasculation is difficult, then you will not get large amount of seed. If it is crop like maize, in backcross you can generate sufficient amount of seed, because you do not have to do any emasculation or even pollination. You just collect plenty of pollen which is available and put it on silk. But in crops like rice or wheat, if you are not having very expert people, developing cross is difficult. If you are going to pulses, again it is more difficult. If you are going to pigeon pea or lentil, seed setting is very poor. So getting a sufficient amount of backcrossed seed is very difficult. Okay, so this is one of the limitation with your backcross population. Now the commonly used, again this is one of the commonly used population, is your recombinant inbred line. What you will do, you will cross resistant parent or parent 1, parent 2, F1, again you test hybridity of F1, then you self that F1, you will get F2, and then F2 you self, you will get F2 derived F3, and then you have to advance from F2 to F6 or later generation. That you can do with single seed descent method. You take one seed from F2 onward. One seed you take from all F2 plant, you will get F3, and then again you take one seed from F3 plant, you will get F4. But if you are taking one seed, what is the problem? That chances are there that you will lose some of the lines. Okay? So if you are starting with 1000 F2 population, you will end up with somewhere 600, 700 your real population. Second thing is there which you should take care. F2 is your most crucial population because each individual is different. So you have to take utmost care that all your F2 seed and F2 plant should survive and they should give sufficient amount of seed. Once you have F2 derived F3, there you will get plenty of seed. So what you can do, in place of one seed, you put 4 seed, 5 seed, or sometime what people will do, they will put complete spike as such. They will not even thresh it, you just put it as such, you will get plenty of plant. Again they will take one spike and they will put complete spike. Or in rice also they are doing, they will put complete panicle. They will harvest one panicle, they will keep panicle intact, then several seedling will grow, and then they will harvest one spike, and then again they will advance. In this way what will happen? Chances of losing a line is less. Surely out of 40, 50 or 100 seed, one spike or one panicle you will get. And once you have achieved homozygosity, then you put it in separate like single seed dibbling you do, and then you harvest from single plant. Okay? So this is going through several cycles of meiosis. So chances of getting recombinant will be high here. Both DH and RIL, they are homozygous. But what is advantage with your RIL? It underwent several cycles of meiosis. So chances are there that you will get some recombinant between your trait of interest and marker. In DH, it went to only one cycle of meiosis. That too in one side only, either in male or female. That is decided by whether you are using maternal haploid or paternal haploid. If you are using pollen culture, so meiosis which has happened in microspore, that only recombinant will go in your DH population. If you are using ovule, the meiosis which has happened in macrospore, that will go to your DH population. Or if you are using maternal haploid inducer line, only the meiosis which is happening on F1 plant on female side, that will be transferred on your DH. So only one side meiosis will be transferred. In RIL, you have both side recombination is getting transferred and you are undergoing several cycle of recombination. So this is the advantage of RIL line over your DH population, although both are homozygous. Okay. Then you have another population is nil. How to develop near isogenic line? It's very easy. You keep on doing back crossing for several generations and recover most of the background of your recurrent parent. So, your P1 recurrent parent and converted line is differing only for few locus, and that one locus will be your gene of interest. So, this will be very useful, especially in case of fine mapping. Fine mapping means you want to identify some co-segregating marker or the marker which is very closely linked. So, you are removing background effect, you are identifying only the marker which is present in that particular region. So, chances are there that you will identify very close markers. Now, once you have developed a phenotyping mapping population, so before mapping you have to go for phenotyping. So, suppose we are trying to map some seedling resistance gene. So, F2 population you inoculate with some pure pathotype of pathogen, then you will be having, if you are using 0 to 4 scale, you will be having different kind of infection type. 0 infection type, you will not be having any uridia and leaf will be completely clean. In FLEC, what you will find, uridia is not there and hypersensitive response will be there. Means you will find hypersensitive response. You have seen, how many of you have seen this plant with hypersensitive response? Okay, those who have seen rust anytime, you must be clear what is hypersensitive response. So, you will find that a small chlorosis has happened on leaf wherever that a spore has fallen, but there is no uridia formation. Okay. Then what you will find, that there is one kind of reaction. There is minute uridia at center and then chlorosis or necrosis is surrounding that area. That surrounding tissue has been killed, but very minute uridia you will find. Then we will be having two kind of reaction and you will be having X kind of reaction. What is X kind of reaction? In a leaf, what you will find sometime, you are having susceptible kind of pustule which is 3 type or 4 type, and you will be having some fleck or one kind of pustule. Yani in same leaf you will find both kind of reaction, the one which is showing resistant and the one which is susceptible. So, we are telling this mesothetic reaction. Again you will find pattern in this mesothetic reaction. What you will find sometime, that this pustule 3 type and fleck type are randomly dispersed in leaf, there is no pattern. Sometime what you will find, at upper tip of leaf hypersensitive response is there and on basal part you are getting 3 kind of reaction, or vice versa. On upper part you are getting 3 kind of reaction and in basal part you are getting fleck kind of reaction. So, based on that you will tell Y and Z kind of reaction, and these 3 and 4 are susceptible kind of pustule. So, once you are phenotyping your F2 population, so if they are giving this kind of reaction, you will put it as resistant, and if that is giving this kind of reaction, you will put as susceptible. So, you will be having in F2 population resistant class or susceptible class, or if you are using F2 derived F3 population, you will be having homozygous resistant class, segregating and homozygous susceptible class. If you are having real population, you will be having homozygous resistant class and homozygous susceptible class. If it is monogenic, it will segregate in 1:1. So, once you have phenotype data, then now you want to map your gene of interest, and the most common way to map your gene is doing bulk segregant analysis. Okay. Now, if you are doing mapping in F2 population, what people will do? They will take 10 to 20 extreme resistant plant and 10 to 20 extreme susceptible plant. You are getting my point? Extract DNA from those 20 or 10 extreme, then add equal amount of DNA from each F2 individual or whatever population you are using. Then you will be having resistant bulk and susceptible bulk. You have parent 1, parent 2, resistant bulk and susceptible bulk. And then, or before this, what some people will do? First, they will do parental polymorphism analysis. They will identify polymorphic marker between parent 1 and parent 2. Suppose you have screened your parents with 1200 SSR marker. Out of 1200, you got some 200 SSR marker which is polymorphic between parent 1 and parent 2. So, after developing bulk, they will screen bulk 1 and bulk 2 with those 200 polymorphic marker, and then they will try to identify which of the marker is putatively linked with your gene of interest. Okay. Can go up to 130, or what is the time? I can go up to 130. Okay. Now you just try to concentrate here. Your gene can be dominant or recessive. Similarly, your marker can be dominant, recessive or codominant. If you are using F2 population, there is some limitation in bulk segregant analysis that will create some problem. Suppose your gene of interest is dominant, your marker is dominant and linkage is in coupling phase. What does it mean? That dominant marker is present with your gene of interest. Capital R is showing amplification of some band and small r is not having any amplification. So, there is presence absence polymorphism and presence is dominant. So, that is called coupling phase. So, resistant parent will be having presence of band, susceptible parent will be having absence of band, resistant bulk will be having presence of band and susceptible bulk will be having absence of band. Here in F2, if we will see resistant bulk, this resistant bulk will be from two kind of individual. One will be homozygous resistant, capital R, that will be resistant, and few individual will be capital R, small r, okay, heterozygote. So, in both the cases, you will get amplification of band. So, here you will get presence absence. So, you can tell that this dominant marker is putatively linked with my gene of interest. Now, coming to another case where you have gene of interest is dominant, marker is recessive and you have repulsion phase linkage. Means your heterozygous individual will be having presence of band. You are getting my point? Here in resistant bulk, you have capital R and capital R, small r. Now, amplification of band is happening because it is linked with small r, and here in bulk you have few individual which is capital R, small r, and that amplification is happening because of those heterozygous individual. Getting my point? So, you will not get any polymorphism although they are linked, but this you can avoid if you are using F2 derived F3 population. You take only homozygous resistant and homozygous susceptible individual. So, you will be having bulk of only capital R here, bulk of small r here. Here you will be having presence of band and here you will be having absence of band. Yes, tell me. First one, that is the easiest one. You have seen gel picture? Okay. Coupling वाला समझ नहीं है. Dominant marker means, what is a dominant trait? In F1, if it is expressing, you will tell that this is a dominant trait. So, suppose you have P1 which is having amplification of band, P2 you have, there is no amplification, absence of band. In F1 it is capital R, small r. So, amplification will happen because of capital R, because marker is linked with capital R. So, in F1 plant you are getting presence of band. So, it is dominant. Okay. So, here it will be presence of band. You are having two kind of individual, capital R and capital R, small r. Both the individual will give you band. So, here you will be having presence of band, and this susceptible bulk is bulk of only small r, which is a recessive class, if you can remember. Okay, so there you will be having absence of band. Here you will be having, in both the cases you will be having presence of band. Here presence has happened because of those heterozygous individual which is present in resistant bulk. Okay. Now coming to recessive gene. If gene of interest is recessive and your marker is dominant and they are linked in repulsion phase, it means small r is having amplification of band. Here what will happen in resistant bulk? You will be having individual small r which are bulked. So here it is presence of band is fine, it should be there. But here in susceptible bulk you are having two kind of susceptible plant. One is homozygous susceptible, another is heterozygous susceptible, capital R, small r. Here again you will get amplification of band because of small r is present in few individual. Okay. Now if it is a recessive gene, molecular marker is recessive and it is in coupling phase, it means your marker is linked with recessive susceptible capital R allele. So here you will be having absence of band. Here even though all the individual are susceptible, two kind of susceptible plant will be there, capital R, small r, you will get a presence of band. So if you are using F2 individual, in these two condition only you can successfully use BSA. Okay. In these two condition you will be having some difficulty. But if you are having co-dominant marker, so there is no question of coupling phase and repulsion phase. Your F1 plant will be having, resistant plant will be having allele 1, susceptible plant will be having allele 2, resistant bulk will be having both the allele if it is bulk from both kind of individual, homozygous resistant and heterozygous resistant. Capital R will give only A type of allele, but suppose out of 10, 2-3 individuals are there, that is heterozygous. So you will get A2 type of allele also. But here in susceptible bulk, that is recessive homozygous class. So here you will get only one type of allele. So sometime people will use only recessive class for mapping. In F2, you generate a large population, take only recessive class and then you do mapping. All this confusion will go. Same thing is there if gene of interest is recessive co-dominant marker. Here in resistant bulk you will be having a specific band compared as of your resistant parent, and your susceptible bulk you will get both kind of band, because here you have both kind of individual, homozygous susceptible and heterozygous susceptible. So if you are having F2 derived F3 family, your work will be very easy. You take homozygous resistant family and you take homozygous susceptible family, bulk that, and then you can go for bulk segregant analysis and you can use any kind of marker, whether that is linked in coupling phase, repulsion phase, that can be used. Now there are different marker system which can be used for genotyping of your mapping population. Initially if you will see during 90s to 2000, around 95 or 2000, there are several paper where people used RAPD marker for mapping of rust resistance gene. What is the RAPD marker? I am sure all of you must be knowing. So they used RAPD marker and then they identified that this particular fragment is linked with your gene of interest. Then they eluted those fragment, got it sequenced, and then they designed primer, and then they got scar markers. What is scar? Sequence characterized amplified region. So sometime what has happened in course of scar development, it lost polymorphism. Then they developed CAPS marker. They used some restriction enzyme, and maybe parent 1 and parent 2 will give you different cleavage pattern. So you will be having different banding pattern, and once SSR marker developed, you will find plenty of papers started coming for mapping of rust resistance gene. Even from IRA, if you will see, several genes has been mapped using scar marker and SSR markers. Dr. Prabhu was there who was working on rust, and he has mapped several of genes like 9, 19, 24, 28. This was resistant to all the known prevalent races in India except one. LR19, only one race is there which is virulent, is 77-7 for LR19. It is 77-8 for LR24. In India, there is no virulent pathotype. And for LR28, 77-7 is there. So, if you are pyramiding these genes, it will be effective. Now, you can use PCR based marker and then you can apply bulk segregant analysis. You identify putative markers and then you genotype your complete F2 population or F2 derived F3 population or whatever population you are using. If you are using RIL, you genotype your RIL population using your polymorphic marker which is there from bulk 1 and bulk 2. And then you go for a scoring and then we will go for some software or analysis like your MapMaker through MapMaker or JoinMap or some other softwares are there, and then you can see what is the linkage pattern and how closely they are linked. Now, with development of your genomic resources, people has developed SNP based markers, okay? And in wheat, these two SNP arrays are most commonly used, 35K and 90K. 35K is from Affymetrix and 90K is from Illumina. So, I will tell I am personally using these two chips. So, I am telling you this will ease your job and you can do the thing which you were doing in 6 to 8 months was required for doing parental polymorphism and bulk segregant analysis. In 20 days you will get it done and you can identify polymorphic marker and then you can convert those polymorphic SNP in PCR or CASP based marker. In previous class he has discussed CASP markers. No? CASP markers, development of CASP marker, okay, let it be. Then you can also use GBS, genotype by sequencing. So, what people will do? You take parent 1, you take parent 2 and take 20 homozygous resistant and 20 homozygous susceptible individual, bulk DNA, and then you go for sequencing of 2 parents and 2 bulks. And then you should go for high coverage, means maybe 20x or 30x. If you are going to more coverage, that will add value to your SNP genotyping. And even you can go for bulk segregant RNA sequencing, that you take P1, P2 and make bulk of 20 homozygous resistant, 20 homozygous susceptible, or in paper you will find some people are using even more individual, or in some paper you will find people are using less individual also. If you are adding more individual, chances are there that some of the SNPs will be missed, but you will get very closely linked SNP. If you are adding less individual, you will get plenty of SNPs and then marker trait association will be difficult. So, you have to optimize the size of the bulk. So, if you will see nowadays, you will find plenty of papers are coming where they are using P1, P2, bulk 1, bulk 2 and they are going to genotyping using SNP array, that may be 35K or 90K. Or if you have money, you can go for genotyping of your whole population, especially if you have RIL, you genotype your complete population. One more thing is there, suppose you have developed a RIL population which is having other traits also which you need to map. Sometime while developing RIL, people will take care that my parent 1 and parent 2 are differing for more than one trait. Like here our target is to map rust resistance gene. Along with rust resistance gene, there are some other genes also, like some seed size genes are there, or some 2 rust resistance gene is there. One is for leaf rust and one is for yellow rust. So, more than one trait is there. In that case, what people will do? They will genotype complete RIL population and then they will go for marker trait association. So, you can map multiple gene in single population. So, here is one example in 2018, this paper published in TAG, and what they did, there is one variety, in short form they are writing it as fried. So, they crossed it with susceptible parent and they developed an F6 population, F6 RIL, and then they developed parent 1, parent 2 and bulk 1 and bulk 2, and they underwent this SNP genotyping using 90K array. So, what they have seen that on 6B there are maximum number of SNPs which is located on 6B, although on other chromosome also SNP is located, but here more than 200 SNPs are present. So, the thing which we were doing through PCR based marker in months, first you have to do parental polymorphism, then you have to do in bulk segregant analysis. Here you just make bulk and you will get what is the location of your gene of interest. Okay. Then sequence information is available, then he has seen what is the region, and then he developed CASP based marker for those SNP, or even you can design normal PCR based marker. Suppose here in P1 and P2, you just take one example. In P1 you have AA, in P2 you have GG. In bulk 1 you have AA, in bulk 2 you have GG. It means they are linked, or suppose this is one of the SNP which is present in this locus. Now, you want to design a primer which amplify your band in resistant parent. So, you take a primer which is complementary to the SNP as in your resistant parent. If you want to design a resistant specific primer, that will be a dominant marker that will show amplification in resistant parent, but not in susceptible parent. But thing is that once you are converting it to PCR based marker, out of 284 only few will give you polymorphism, because only one base pair difference sometime in amplification that will be overruled. So, you have to design in such a way that that SNP can be converted to PCR based marker. And then with this bulk you can tell one more thing you have to take care, that with this bulk you will come to know that these are the SNP which is present in resistant bulk and polymorphic to susceptible bulk. But what is the distance, that you cannot tell, because for calculating distance you have to run that SNP in whole population, and then based on recombination frequency you can tell that this is the distance. So, for calculating distance you have to design either CASP marker or PCR based marker, and then you can calculate that this SNP is located at this much distance to my gene of interest. Okay. But you can design very closely linked marker and very rapidly within 1 or 2 months, and your price will be saved. Here only 4 sample, or even if you are going to replicate 8 sample, P1, P2, bulk 1, bulk 2, you put in 2 replication. Okay. So, that you will be sure that whatever polymorphism you are getting, that is confirmed twice. So, you will be able to identify very easily within 20-30 days, and 8 sample will cost, if you are going for 35K, it will cost not more than 35 to 40,000. If you are going to 90K, it will cost 60 or something like that. So, that is cheap, and very rapidly you can tell this is my location of gene. And second thing which I discussed earlier, that if you are knowing that in this region already 4 genes are reported, okay, then rapidly you can see, okay, whether my gene of interest is one among already known or this is novel. Sometime based on this SNP location you can tell that if suppose for other 3 gene locus is known that this is present here, and here my gene of interest is present, so their location is different, that is why my gene of interest is different, or through test of allelism also you can tell that my gene of interest is different. Another is your GBS, genotype by sequencing. Again they will go for genotyping using sequencing. Here you should go for more coverage, and you will calculate SNP index. Suppose 20x coverage you are taking. So, whether all 20x coverage or all 20 is having SNP as of your resistant parent, all 20 is having as of your susceptible parent, or out of 20, 2 SNP is similar to susceptible parent and 18 is similar to resistant parent. So, your SNP index can be calculated like 2 by or 18 by 20, it will be 0.9. If it is all 20 are resistant specific, SNP index will be 1. Now, another approach is your bulk segregant RNA-seq. You can take RNA from 20 resistant bulk and 20 susceptible bulk, and then you can go for RNA sequencing, and then again you can see what is the polymorphic between parent 1, parent 2, and bulk of 20 resistant individual and bulk of 20 susceptible individual, and then you can identify SNPs which is co-localized or which is present in your two different bulk, and then you can design PCR based marker for this. For all the techniques you will find, especially if you will read tag, in all the issue you will find one or other paper for rust resistance, and they are using GBS or SNP genotyping or RNA-seq. If you will find tag, molecular breeding, this kind of general civil search, you will keep on finding that every week or every month one new gene or something is coming. So, ultimately why we are doing all this? Why you want to map or why you want to have molecular marker? We can transfer our resistance gene even. इसको बंद करने का कहीं है? Okay, why you want to have marker? You can transfer your resistance gene even conventionally. Simply you cross your resistant parent with susceptible parent and you transfer it through back crossing or simply by selfing and selecting resistant plant. But for selection of resistant plant, first thing which is required is you have to do phenotyping. Okay? So, sometime what will happen? Plant will look resistant because of escape. So, if you are doing transfer through conventional method, you have to ensure that your disease development is proper, and whatever plant is coming resistant is because of resistant gene, not because of escape. But if you have molecular marker linked with your gene of interest, you can transfer with high confidence that my marker is present. It means that resistance gene should be present. Second thing, if you want to pyramid two genes for which virulent race are not known, in that case presence of both the gene cannot be confirmed phenotypically. You are getting my point? Suppose you want to pyramid LR24 and LR42. For both the gene there is no virulence. So, even if single gene is present, that will be resistant to all the pathotype, either LR24 or LR42, or if both the genes are there, then also it will be resistant to all the pathotypes. So, how to identify those individual which is carrying both the genes? This is possible if you have marker, that you see whether marker linked to both the genes are amplifying, it means my individual is having both the genes. Third thing will come if you want to pyramid one seedling resistant gene and one adult plant resistant gene. Presence of seedling resistance gene mask your APR gene. Plant will be resistant because of presence of that seedling gene. Maybe in background APR gene is present. So, if you have molecular marker linked with APR gene, so you can see that what are the individuals which is carrying your adult plant resistance gene. Conventional method can be used. If you are adding one seedling resistance gene and one APR gene, conventional method can be used. How you can use? Suppose I want to pyramid one seedling resistance gene which is LR9 and one APR gene which is LR34. Now, we have a virulent race against LR9. Okay? First you test that whether my plant is carrying seedling resistance gene or not. How you will test? Take one avirulent gene like avirulent pathotype like 77-5, you test, okay, my seedling is having LR9. Now, that LR9 is not effective against 77-7. So, in field you screen against 77-7. So, presence of APR you can detect with virulent pathotype which is virulent for your seedling resistance gene. But again that is difficulty, in field maintaining purity is difficulty. So with help of molecular marker, you can very easily pyramid one seedling resistance gene and one adult plant resistance gene. This is all which I wanted to talk. If you want to ask anything, you can ask, or it's already 1:15, I don't know. Or if even if you want to ask anything later on, I'm in genetics division. Today afternoon we have one practical, so there you can ask. Or even if later on after going to your college you want to ask, you can mail me. I think my mail address will be there in that booklets. Okay, all the best. How many of you are working on Rust? Any one of you? One, two, wheat Rust, you are working on wheat Rust? Okay. You are from? Afghanistan. So you came for this training or you are in some other training? Okay, you are in HA. I'll tell you, Rust, doing genetics on Rust is a wonderful thing. Every day you will be happy to see your plants and all these things which you are telling, 3:1, 15:1, or whatever ratio, you will see by yourself that this is happening. And you will feel amazed if you will work that. And very easily you can see that resistance is happening or not, how it is getting bred, how two pathotypes are there. If you will maintain purity of pathotypes, with one pathotype you will see that I am getting fleck reaction. The moment you will inoculate with second pathotype which is virulent, you will see three kind of pustules coming. So, and everything will be visible from your eyes. You don't need to predict anything that because of this something has happened, because of that something has happened. If you are following protocol, if your inoculum is viable, if your plant is true to the type, if you are maintaining purity, you will be happy to see your work every day. Okay.